DNA replication.
DNA replication is the process of producing two identical replicas from one original DNA molecule. This biological process occurs in all living organisms and is the basis for biological inheritance. DNA is made up of two strands, and each strand of the original DNA molecule serves as template for the production of the complementary strand, a process referred to as semiconservative replication. Cellular proofreading and error-checking mechanisms ensure near-perfect fidelity for DNA replication. In a cell, DNA replication begins at specific locations, or origins of replication, in the genome. Unwinding of DNA at the origin and synthesis of new strands results in replication forks growing bidirectional from the origin. A number of proteins are associated with the replication fork, which helps in terms of the initiation and continuation of DNA synthesis. Most prominently, DNA polymerase synthesizes the new DNA by adding complementary nucleotides to the template strand. DNA replication can also be performed in vitro, artificially, outside a cell. DNA polymerase is isolated from cells, and artificial DNA primers can be used to initiate DNA synthesis at known sequences in a template DNA molecule. The polymerase chain reaction, PCR, a common laboratory technique, cyclically applies such artificial synthesis to amplify a specific target DNA fragment from a pool of DNA.
Background on DNA structure.
DNA usually exists as a double-stranded structure, with both strands coiled together to form the characteristic double helix. Each single strand of DNA is a chain of 4 types of nucleotides. Nucleotides in DNA contain a deoxyribose sugar, a phosphate, and a nucleobase. The 4 types of nucleotide correspond to the 4 nucleobases adenine, cytosine, guanine, and thymine, commonly abbreviated as A, C, G and T. Adenine and guanine are purine bases, while cytosine and thymine are pyrimidines. These nucleotides form phosphordiester bonds, creating the phosphate deoxyribose backbone of the DNA double helix with the nucleobases pointing inward. Nucleotides, bases, are matched between strands through hydrogen bonds to form base pairs. Adenine pairs with thymine, 2 hydrogen bonds, and guanine pairs with cytosine, stronger, 3 hydrogen bonds. DNA strands have a directionality, and the different ends of a single strand are called the 3' end, and the 5' end. By convention, if the base sequence of a single strand of DNA is given, the left end of the sequence is 5' end, while the right end of the sequence is the 3' end. The strands of the double helix are anti-parallel, with one being 5' to 3', and the opposite strand 3' to 5'. These terms refer to the carbon atom in deoxyribose to which the next phosphate in the chain attaches. Directionality has consequences in DNA synthesis, because DNA polymerase can synthesize DNA in only one direction by adding nucleotides to the 3' end of a DNA strand. The pairing of complementary bases in DNA through hydrogen bonding means that the information contained within each strand is redundant. The nucleotides on a single strand can be used to reconstruct nucleotides on a newly synthesized partner strand.
DNA polymerase.
DNA polymerases are a family of enzymes that carry out all forms of DNA replication. DNA polymerases in general cannot initiate synthesis of new strands, but can only extend an existing DNA or RNA strand paired with a template strand. To begin synthesis, a short fragment of RNA, called a primer, must be created and paired with the template DNA strand. DNA polymerase synthesizes a new strand of DNA by extending the 3' end of an existing nucleotide chain, adding new nucleotides matched to the template strand one at a time via the creation of phosphordiester bonds. The energy for this process of DNA polymerization comes from hydrolysis of the high-energy phosphate, phosphoanhydride, bonds between the 3 phosphates attached to each unincorporated base. Free bases with their attached phosphate groups are called nucleotides. In particular, bases with 3 attached phosphate groups are called nucleoside trophosphates. When a nucleotide is being added to a growing DNA strand, the formation of a phosphordiester bond between the proximal phosphate of the nucleotide to the growing chain is accompanied by hydrolysis of a high-energy phosphate bond with release of the two distal phosphates as a pyrophosphate. Enzymatic hydrolysis of the resulting pyrophosphate into inorganic phosphate consumes a second high-energy phosphate bond and renders the reaction effectively irreversible. In general, DNA polymerases are highly accurate, with an intrinsic error rate of less than 1 mistake for every 107 nucleotides added. In addition, some DNA polymerases also have proofreading ability. They can remove nucleotides from the end of a growing strand in order to correct mismatched bases. Finally, post-replication mismatch repair mechanisms monitor the DNA for errors, being capable of distinguishing mismatches in the newly synthesized DNA strand from the original strand sequence. Together, these three discrimination steps enable replication fidelity of less than 1 mistake for every 109 nucleotides added. The rate of DNA replication in a living cell was first measured as the rate of phage T4 DNA elongation in phage-infected E. coli. During the period of exponential DNA increase at 37 DEGC, the rate was 749 nucleotides per second. The mutation rate per base pair per replication during phage T4 DNA synthesis is 1.7 per 10^8. Thus DNA replication is both impressively fast and accurate.
Replication process.
DNA replication, like all biological polymerization processes, proceeds in three enzymatically catalyzed and coordinated steps: initiation, elongation and termination.
Initiation.
For a cell to divide, it must first replicate its DNA. This process is initiated at particular points in the DNA, known as origins, which are targeted by initiator proteins. In E. coli this protein is DNA. In yeast, this is the origin recognition complex. Sequences used by initiator proteins tend to be AT-rich, rich in adenine and thymine bases, because AT base pairs have two hydrogen bonds, rather than the three formed in a CG pair, which are easier to unzip. Once the origin has been located, these initiators recruit other proteins and form the pre-replication complex, which unzips the double-stranded DNA.
Elongation.
All known DNA replication systems require a free 3' hydroxyl group before synthesis can be initiated. Important note: DNA is read in 3' to 5' direction, whereas a new strand is synthesized in the 5' to 3' direction. This is often confused. 4 distinct mechanisms for synthesis have been described. All cellular life forms and many DNA viruses, phages and plasmids use a primers to synthesize a short RNA primer with a free 3' OH group, which is subsequently elongated by a DNA polymerase. The retroelements, including retroviruses, employ a transfer RNA that primes DNA replication by providing a free 3' OH that is used for elongation by the reverse transcriptase. In the adenoviruses and the penthouse 29 family of bacteriophages, the 3' OH group is provided by the side chain of an amino acid of the genome-attached protein, the terminal protein, to which nucleotides are added by DNA polymerase to form a new strand. In the single-stranded DNA viruses, a group that includes the circoviruses, the geminoviruses, the parvoviruses and others, and also the many phages and plasmids that use the rolling circle replication (RCR) mechanism, the RCR endonuclease creates a nick in the genome strand. Single-stranded viruses, or one of the DNA strands, plasmids. The 5' end of the nicked strand is transferred to a tyrosine residue on the nucleus, and the free 3' OH group is then used by DNA polymerase for new strand synthesis. The first is the best known of these mechanisms and is used by the cellular organisms. In this mechanism, once the two strands are separated, primers adds RNA primers to the template strands. The leading strand receives one RNA primer while the lagging strand receives several. The leading strand is continuously extended from the primer by a high processivity, replicative DNA polymerase, while the lagging strand is extended discontinuously from each primer, forming Okazaki fragments. RNase removes the primer RNA fragments, and a low processivity DNA polymerase distinct from the replicative polymerase enters to fill the gaps. When this is complete, a single nick on the leading strand and several nicks on the lagging strand can be found. Ligase works to fill these nicks in, thus completing the newly replicated DNA molecule. The primers used in this process differs significantly between bacteria and Archaea eukaryotes. Bacteria use a primus belonging to the DNA G protein superfamily which contains a catalytic domain of the top rim fold type. The top rim fold contains an AB core with 4 conserved strands in a Rossmann-like topology. This structure is also found in the catalytic domains of topoisomerase I, topoisomerase II, the OLD family nucleases and DNA repair proteins related to the RECR protein. The primus used by Archaea and eukaryotes in contrast contains a highly derived version of the RNA recognition motif, RRM. This primus is structurally similar to many viral RNA-dependent RNA polymerases, reverse transcriptases, cyclic nucleotide-generating cyclases and DNA polymerases of the ABY families that are involved in DNA replication and repair. All these proteins share a catalytic mechanism of D-metal ion-mediated nucleotide transfer, whereby 2 acidic residues located at the end of the first strand and between the second and third strands of the RRM-like unit respectively, chelate 2 divalent cations. Multiple DNA polymerases take on different roles in the DNA replication process. In E. coli, DNA pol3 is the polymerase enzyme primarily responsible for DNA replication. It assembles into a replication complex at the replication fork that exhibits extremely high processivity, remaining intact for the entire replication cycle. In contrast, DNA pol-I is the enzyme responsible for replacing RNA primers with DNA. DNA pol-I has a 5' to 3' exonuclease activity in addition to its polymerase activity, and uses its exonuclease activity to degrade the RNA primers ahead of it as it extends the DNA strand behind it, in a process called nick translation. Pol-I is much less processive than pol3 because its primary function in DNA replication is to create many short DNA regions rather than a few very long regions. In eukaryotes, the low processivity initiating enzyme, pol-A, has intrinsic primus activity. The high processivity extension enzymes are pol-D and pol-E. As DNA synthesis continues, the original DNA strands continue to unwind on each side of the bubble, forming a replication fork with 2 prongs. In bacteria, which have a single origin of replication on their circular chromosome, this process creates a theta structure, resembling the Greek letter theta, th. In contrast, eukaryotes have longer linear chromosomes and initiate replication at multiple origins within these.
Replication fork.
The replication fork is a structure that forms within the nucleus during DNA replication. It is created by helicases, which break the hydrogen bonds holding the 2 DNA strands together. The resulting structure has 2 branching prongs, each one made up of a single strand of DNA. These 2 strands serve as the template for the leading and lagging strands, which will be created as DNA polymerase matches complementary nucleotides to the templates. The templates may be properly referred to as the leading strand template and the lagging strand template. DNA is always synthesized in the 5' to 3' direction. Since the leading and lagging strand templates are oriented in opposite directions at the replication fork, a major issue is how to achieve synthesis of nascent, new, lagging strand DNA, whose direction of synthesis is opposite to the direction of the growing replication fork.
Leading strand.
The leading strand is the strand of nascent DNA which is being synthesized in the same direction as the growing replication fork. A polymerase reads the leading strand template and adds complementary nucleotides to the nascent leading strand on a continuous basis. The polymerase involved in leading strand synthesis is DNA polymerase III, DNA pol3, in prokaryotes and presumably pol-E in yeasts. In human cells, the leading and lagging strands are synthesized by pol E and pol D, respectively, within the nucleus, and pol G in the mitochondria. Pol E can substitute for pol D in special circumstances.
Lagging strand.
The lagging strand is the strand of nascent DNA whose direction of synthesis is opposite to the direction of the growing replication fork. Because of its orientation, replication of the lagging strand is more complicated than that of the leading strand. The lagging strand is synthesized in short, separated segments. On the lagging strand template, a primus reads the template DNA and initiates synthesis of a short complementary RNA primer. A DNA polymerase extends the primed segments, forming Okazaki fragments. The RNA primers are then removed and replaced with DNA, and the fragments of DNA are joined together by DNA ligase. In eukaryotes, primus is intrinsic to pol A. DNA polymerase III, in prokaryotes, or pol D pol E, in eukaryotes, is responsible for extension of the primed segments. Primer removal in eukaryotes is also performed by pol D. In prokaryotes, primer removal is performed by DNA polymerase I, which reads the fragments, removes the RNA using its flap endonuclease domain. RNA primers are removed by 5-3 exonuclease activity of polymerase I, and replaces the RNA nucleotides with DNA nucleotides.
Dynamics at the replication fork.
As helicase unwinds DNA at the replication fork, the DNA ahead is forced to rotate. This process results in a buildup of twists in the DNA ahead. This buildup forms a torsional resistance that would eventually halt the progress of the replication fork. DNA gyrase is an enzyme that temporarily breaks the strands of DNA, relieving the tension caused by unwinding the two strands of the DNA helix. DNA gyrase achieves this by adding negative supercoils to the DNA helix. Bare single-stranded DNA tends to fold back on itself, forming secondary structures. These structures can interfere with the movement of DNA polymerase. To prevent this, single-strand binding proteins bind to the DNA until a second strand is synthesized, preventing secondary structure formation. Clamp proteins form a sliding clamp around DNA, helping the DNA polymerase maintain contact with its template, thereby assisting with processivity. The inner face of the clamp enables DNA to be threaded through it. Once the polymerase reaches the end of the template or detects double-stranded DNA, the sliding clamp undergoes a conformational change that releases the DNA polymerase. Clamp loading proteins are used to initially load the clamp, recognizing the junction between template and RNA primers 274-5.
DNA replication proteins.
At the replication fork, many replication enzymes assemble on the DNA into a complex molecular machine called the replisome. The following is a list of major DNA replication enzymes that participate in the replisome.
Termination.
Eukaryotes initiate DNA replication at multiple points in the chromosome, so replication forks meet and terminate at many points in the chromosome. These are not known to be regulated in any particular way. Because eukaryotes have linear chromosomes, DNA replication is unable to reach the very end of the chromosomes, but ends at the telomere region of repetitive DNA close to the end. This shortens the telomere of the daughter DNA strand. Shortening of the telomeres is a normal process in somatic cells. As a result, cells can only divide a certain number of times before the DNA loss prevents further division. This is known as the Hayflick limit. Within the germ cell line, which passes DNA to the next generation, polymerase extends the repetitive sequences of the telomere region to prevent degradation. Polymerase can become mistakenly active in somatic cells, sometimes leading to cancer formation. Termination requires that the progress of the DNA replication fork must stop or be blocked. Termination at a specific locus, when it occurs, involves the interaction between two components, 1, a termination site sequence in the DNA, and 2, a protein which binds to this sequence to physically stop DNA replication. In various bacterial species, This is named the DNA replication terminus site binding protein, or to protein. Because bacteria have circular chromosomes, termination of replication occurs when the two replication forks meet each other on the opposite end of the parental chromosome. E. coli regulates this process through the use of termination sequences that, when bound by the two's protein, enable only one direction of replication fork to pass through. As a result, the replication forks are constrained to always meet within the termination region of the chromosome.
Regulation. Eukaryotes.
Within eukaryotes, DNA replication is controlled within the context of the cell cycle. As the cell grows and divides, it progresses through stages in the cell cycle. DNA replication occurs during the S phase, synthesis phase. The progress of the eukaryotic cell through the cycle is controlled by cell cycle checkpoints. Progression through checkpoints is controlled through complex interactions between various proteins, including cyclins and cyclin-dependent kinases. The G1 per second checkpoint, or restriction checkpoint, regulates whether eukaryotic cells enter the process of DNA replication and subsequent division. Cells that do not proceed through this checkpoint remain in the GO stage and do not replicate their DNA. Replication of chloroplast and mitochondrial genomes occurs independently of the cell cycle, through the process of D-loop replication.
Bacteria.
Most bacteria do not go through a well-defined cell cycle but instead continuously copy their DNA. During rapid growth, this can result in the concurrent occurrence of multiple rounds of replication. In E. coli, the best characterized bacteria, DNA replication is regulated through several mechanisms, including the hemimethylation and sequestering of the origin sequence, the ratio of adenosine triphosphate, ATP, to adenosine diphosphate, ADP, and the levels of protein DNA. All these control the binding of initiator proteins to the origin sequences. Because E. coli methylates GATC DNA sequences, DNA synthesis results in hemimethylated sequences. This hemimethylated DNA is recognized by the protein Sec-A, which binds and sequesters the origin sequence. In addition, DNA-A, required for initiation of replication, binds less well to hemimethylated DNA. As a result, newly replicated origins are prevented from immediately initiating another round of DNA replication. ATP builds up when the cell is in a rich medium, triggering DNA replication once the cell has reached a specific size. ATP competes with ADP to bind to DNA-A, and the DNA-A-ATP complex is able to initiate replication. A certain number of DNA-A proteins are also required for DNA replication. Each time the origin is copied, the number of binding sites for DNA-A doubles, requiring the synthesis of more DNA-A to enable another initiation of replication.
Polymerase chain reaction.
Researchers commonly replicate DNA in vitro using the polymerase chain reaction, PCR. PCR uses a pair of primers to span a target region in template DNA, and then polymerizes partner strands in each direction from these primers using a thermostable DNA polymerase. Repeating this process through multiple cycles produces amplification of the targeted DNA region. At the start of each cycle, the mixture of template and primers is heated, separating the newly synthesized molecule and template. Then, as the mixture cools, both of these become templates for annealing of new primers, and the polymerase extends from these. As a result, the number of copies of the target region doubles each round, increasing exponentially.
Notes.
The energetics of this process may also help explain the directionality of synthesis. If DNA was synthesized in the 3 feet to 5 feet direction, the energy for the process would come from the 5 feet end of the growing strand rather than from free nucleotides. The problem is that if the high-energy triphosphates were on the growing strand and not on the free nucleotides, proofreading by removing a mismatched terminal nucleotide would be problematic. Once a nucleotide is added, the triphosphate is lost and a single phosphate remains on the backbone between the new nucleotide and the rest of the strand. If the added nucleotide were mismatched, removal would result in a DNA strand terminated by a monophosphate at the end of the growing strand rather than a high-energy triphosphate. So strand would be stuck and wouldn't be able to grow anymore. In actuality, the high-energy triphosphates hydrolyzed at each step originate from the free nucleotides, not the polymerized strand, so this issue does not exist.